Today we will study about the histological structure of the striated muscle. This is one of the muscular tissue known as striated muscles. It consists of all the uh, chemistry or the all the constituents similar to that of the muscular tissue. So here our topic is nothing but the striated muscles. These muscles show cross striation under the microscope, hence the name. They are also called skeletal or voluntary muscles. This tissue originates from embryonic myoblast of the mesoderm. It consists of very long, about 1 to 40 millimeter, and uniformly cylindrical multinucleate myofibers about 10 to 100 micrometer or more in diameter. Each striated muscle fiber or cell is enclosed by the sarcolemma. Electron microscopy shows it consists of plasma lemmas supported by a basal lamina and fine reticular fibers of collagen. Inside the sarcolemma are the nuclei and myofibrils. Around and among the fibrils and accumulated near the nuclei is the fluid known as sarcoplasm. It contains numerous and large mitochondria, small Golgi complex, non-granular cytoplasmic reticulum, and few ribosomes. In addition, glycogen granules and some lipid droplets also occur. The mitochondria are concerned with ATP production, which provides energy required for muscle contraction. Function of the sarcoplasmic reticulum is to make calcium ions available for contraction of the muscle fibers. In an average sized fibers, there are several hundred nuclei. They are flattened, elongated, oval bodies located peripherally just under the sarcolemma. The sarcolemma contains compact bundles of numerous cylindrical and thin myofibrils lying parallel to each other. Each myofibril is composed of two types of regularly alternating bands of dark and light material, giving it a cross striated appearance. One type is the anisotropic or A band, while the another type is isotropic or I bands, which are alternating present. In a muscle fiber, the myofibrils show a transverse dark and light bands. In resting fibrils, both the bands are of equal length. When the fibril contracts, the I bands shorten, while it is stretched, the I bands appear longer than the A bands. Thus, the A bands always remain unchanged in length. Each of the A and I bands appear bisected. Across the A band is a lighter Hensen's line or H line. H, also called healer, means lighter. Similarly, the I band is traversed by causes membrane, which is more familiarly called Z line or Z disc. Z is nothing but the Zwischen's chip, meaning intermediate disc. The unit of length between two Z discs is called sarcomere. This is also the functional unit of the striated muscle. Each myofibril consists of repeating array of sarcomeres, each about 2 micron long in resting muscles. The sarcolemma invaginates deeply at the Z line to form transverse sarcotubules or T tubules. These are useful in the transmission of impulses through the muscle fibers. Electron microscopy has shown that each sarcomere contains longitudinally oriented proteinous myofilaments of two kinds: primary or thick filament composed of myosin, and secondary or thin filament containing actin. The myosin filaments are about 1.6 micron long and nearly 15 nanometer thick. They are restricted to the central position of the sarcomere, that is in the A band. The actin filaments are about 1 micron long and 7 nanometer in diameter. They occur in the I band, extend for some distance into the A band. There are approximately 1500 myosin and 3000 actin filaments in a myofibril. They are the contractile element in the muscle cells. It is now universally accepted that the contraction of muscle cell is effected by a sliding filament mechanism. For operation of this process, it is essential that one end of each actin filament is immobilized. Thus, in a striated muscle, actin filament is fixed to the Z line. In smooth muscle, cytoplasmic plaque seem to serve the same anchoring function. The cardiac muscle fiber has terminal structures called intercalated discs adjacent to the sarcolemma, and actin filaments are attached to these discs. In striated muscle cells, one end of each filament is anchored to the Z disc by means of protein cap Z. Alpha-actinin and vinsulin. The free portion of actin filament extend towards the center of the sarcomere and interdigitate with the myosin filaments. Associated with the free portion of the actin filaments are the proteins tropomyosin and troponins. Like actin and myosin, these two proteins are also involved in the process of muscle contraction. During the contraction and relaxation of the striated muscle, the myosin filaments must be kept centered in the sarcomere. This function seems to be carried out by an elastic fibrous protein called tintin, which attach the myosin filament to the Z disc. Extending from the Z disc into the sarcomere are filaments of another protein termed nibulin. This inelastic protein is involved in the formation of actin filaments. It may also organize the actin filaments into a regular geometrical pattern relative to the myosin filaments. Thus, titin and nibulin appear to impart structural stability to the sarcomeral contractile machinery. The mechanism of contraction and relaxation of the muscle fiber occurs due to the actin and myosin filaments, as it has been shown by H.E. Huxley. The contraction of sarcomere and hence of muscle fiber does not involve shortening of either the primary or secondary filaments. Contraction of the sarcomere is due to the actin filament sliding deeper and deeper between the myosin filaments, pulling along the Z disc to which they are attached until at full contraction the free ends of the secondary filaments meet in the middle of the sarcomere. Because of this, the Z discs are brought closer together and sarcomere is thus shortened. This is generally termed the sliding filament mechanism of muscle contraction, in which the actin and myosin filament interact to form cross bridge, which are seen in the figure. So, the contraction of the muscle fiber is in response to motor impulses from the central nervous system. In a contracting fiber, ATP is broken down to ADP, releasing a lot of energy. This reaction is catalyzed by myosin. The resultant energy is used for the myosin and actin filaments to slide against each other, thereby shortening the sarcomeres and contracting the muscle fiber. Presence of calcium is essential for this process. So this is about the